Hi, good evening. Myself, K. Satish Kumar. I have completed my MSc Zoology, SP University. Now my topic is Innate Immunity and Acquired Immunity, the types of immunity. Next, immunity. It is a response that is generated by a body towards a disease-causing pathogen. Now, the study of or the branch of science that is dealing with the study of immunity and immune system is called as immunology. The father of immunology is Edward Jenner, whereas the modern father of immunology is Louis Pasteur. As we know the great significance of those scientists who have contributed themselves for the development of immune system and immunology. Now, coming to the immunity, we need to know about two basic things like antigen and antibody. Antigen, it is a foreign substance which promotes the producing of antibodies. Coming to antibodies, what is meant by an antibody? Antibody is a structure which is developed in our body which will be killing the disease causing organisms. This is a Y-shaped organelle. Now, the structure of this antibody was given by Robert Porter. He is a scientist who gave this basic structure of the antigen. Now, in this Y-shaped antigen structure, it has two heavy chains and two light chains. These heavy chains and light chains are linked by disulfide bonds. At the opening of the Y shape, you can see an FAB end, fragment antigen binding site. Whereas at the bottom you have FC end, fragment cell binding ends. These ends are responsible for capturing an antigen. Now, going to our topic is immunity. Innate immunity and acquired immunity. The name itself, innate immunity, says that it is by birth, or it's when an organism gave birth that has developed some type of mechanisms in their body to prevent from disease-causing organisms by naturally. Now, these will be developed by these organisms at the time of birth. They will be present inside their body, and these are non-specific type of defense. That means they won't even have a prior contact of microorganisms. They have developed naturally in their body. Next, coming to this, they have some different types of barriers to prevent these microorganisms or pathogens to enter into our body. Like 4 barriers: physical barrier, physiological barrier, cellular barrier and cytokine barriers. These are the 4 major types of barriers that are developed in the innate immunity. Coming with the first one is physical barrier. Best example for physical barrier is skin and mucus. Skin and mucus acts as a great physical barriers. Skin, you know about skin. Skin is the outermost and largest organ present in an organism and which is highly exposed to the outer environment. You know, very more amount of microorganisms are growing in the environment. These will be getting contacted with the skin, but it is not even allowing each and every microorganism to enter into our body. Next is mucus. Mucus is present in your nasal cavities and nasal chambers, and they will be responsible for trapping of the microorganisms and will not be allowing them to enter into our body. So that's why this physical barrier acts like a first line of defense in your body. Next, coming to our physiological barrier. This acts like a second line of defense where the digestive juices and the sweat, saliva, tear which is secreted from the eyes acts as a physiological barriers. You know, whatever the food we are taking, they do contain microorganisms. When we are eating those foods, the HCl which is present in our stomach will kill those microorganisms and will purify the food whichever we are eating. Next, the tears and the sweat and the saliva, they contain a major enzyme called as lysozyme. This lysozyme enzyme is responsible for breaking the bacterial cell wall peptidoglycan. This peptidoglycan cell wall will be ruptured by this physiological barriers. It acts like the second line of defense. Next, cellular barriers. like macrophages, monocytes, neutrophils. These cells which are present in our blood supply, they will phagocyte and they'll kill the microorganisms whichever they are entering into our body, which acts like a third line of defense. Next, coming to the cytokine barriers. Cytokine barriers are responsible for protecting the non-infected cells from the infected cells. If suppose if you have an infection, if it is spreading overly, with the help of the cytokine barriers, the infected cells and the non-infected cells will be differentiated. The non-infected cells will not be affected by the infected cells with the help of this cytokine barriers. Next is acquired immunity. The name itself represents the acquired immunity, means after birth. The immunity that is developed by an organism after the birth of an organism. Like, if you go with the types of this acquired immunity, this is two types: active acquired immunity and passive acquired immunity. See, this is a pathogen specific immunity. That means when the body is contacted with a microorganism, then only the body will develop the antibodies. So that type of immunity is called as active acquired immunity. Let's come into the types: active acquired immunity and passive acquired immunity. Active acquired immunity. In this, the immunity that is developed by the introduction or else the production of antibodies after the infection is called as active acquired immunity. In this, it is again two types: natural active acquired immunity and artificial active acquired immunity. First, natural active acquired immunity. Suppose when a person is infected with a disease, let's take an example of smallpox and chickenpox. After a few days, the person will get automatically cured without any medication. That means that antibodies will be produced in the body. So that type of immunity is called as natural active acquired immunity. Next is artificial active immunity. In this, the immunity that is developed by induction of weakened antibodies or antigens of a disease causing organisms. Suppose if you know about this corona vaccine, you know about everyone have the corona vaccine. That means the heat killed virulent or else less weakened organism which is causing the disease will be inserted into your body, and that weakened antigen will produce the antibodies which are responsible for killing that particular microorganism. So that type of immunity is called as artificial active acquired immunity through vaccination. It is through vaccination. Next is passive acquired immunity. In this, the ready-made antibodies will be inserted into an individual and it will be developing the immunity. So that type of immunity is called as passive acquired active immunity. Next, coming to the types: natural passive immunity and artificial passive immunity. Natural passive immunity, you know the relationship between a mother and child, right? See, the mother is responsible for the entire development of the baby. In the same manner, for the development of antibodies also, this IgG, the major antibody IgG, will be transferred from mother to the children through placenta, and one more antibody is IgA. It will be transferred from colostrum through milk. So these two are the major antibodies that will be transferred, which are naturally produced in the mother's body and will be entering into the child body. So that type of immunity is called as natural passive acquired immunity. Next, coming to artificial passive immunity. In this, the preformed antibodies, like if you take the snake bite, venom of a snake bite, anti-venom, anti-serum for tetanus, anti-serum for rabies, these will be produced by injecting those organisms, microorganisms, into the organisms like horse or sheep. So that type of immunity is called as artificial passive immunity. Now, coming to the responses, there is primary response and secondary response. Primary response: whenever a microorganism enters into our body, not only microorganism, any pathogen, if enter into our body, then the response that is shown by our body is called as primary response. Next, coming to secondary response. Now, whatever the microorganism which is entered previously, the information will be stored in our memory cells. Now, if in future, if the same microorganism, if it enters into our body, then particular type of antibodies will be produced and it will be completely killed here itself without a fraction of times. Now, coming to this, based on the types of responses, we have two mechanisms, that is humoral immunity and cell-mediated immunity, through which the antibodies will be phagocytes or killed. Now, coming to humoral immunity, in this, the fluid part, the lymph and plasma plays an important role in this humoral immunity. Whenever an antigen enters into our body, take an example, some bacteria has entered into our body. Now, the antigens that are related to this particular bacteria will be represented by the FAB ends, fragment antigen binding ends. They will display the information on the class 2 MHC, major histocompatibility complex. After displaying the information of this particular antigen which is entered into our body, that information will be gathered by these cells and they will produce the interleukins. See, the interleukins are the major important thing which are responsible for producing of memory cells and plasma cells. Memory cells, you know about memory, right? These memory cells will store the information of that particular antigen. If it is of a star shape, that information will be stored. If it is circular shape, the information will be stored. So based on the shape, the information will be stored on the memory cells. Now, we have plasma cells. You know, whatever the shape that is present in the antigen, that opposite type of antibodies will be produced by this plasma cells. And when these are produced, these antibodies will act on those antigens and they will agglutinate. They will be causing agglutination and precipitation of soluble antigens and they will be killing them by phagocytosis. This is how the humoral immunity takes place. Now coming to the next one, cell-mediated immunity. In this, the cell-mediated immunity, the T cells and natural killer cells which are called as T cytotoxic cells or NK cells, they play the important role for mediating this immunity. Now, next major thing is APCs and ASCs, antigen presenting cells and altered cells. We have class 2 MHC humoral immunity, whereas coming to in cell-mediated immunity, this APCs and ASCs plays an important role for displaying the information of this particular antigen which is mediated by the cells, natural killer cells or T cells. Now, after displaying the information, the T cells will differentiate into T memory cells and after that, the T cells will release as interleukins. The T memory cells will save the information, whereas the TH cells, called the T helper cells, they will produce the interleukins and these interleukins in turn, they will be producing perforins and granozymes. Perforins, the name itself, perforation, that means they cause holes to the cell. After causing the holes to the cell, the granozymes will enter into those holes and they'll kill the bacteria and microorganisms which are present in the cell. And then, which causes the apoptosis, that is nothing but cell death. This is how the cell-mediated immunity takes place. Thank you.